This award in diffraction physics was established in 1970 by students and friends of Professor Bertram E. Warren on the occasion of his retirement from the Massachusetts Institute of Technology. The award seeks to recognize an important recent contribution to the physics of solids or liquids using X-ray, neutron, or electron diffraction techniques. I should say and/or in this case. The 2020 Warren awardee is Professor Jacqueline M. Cole, head of the Molecular Engineering Group at Cavendish Laboratory at the University of Cambridge. What the Brits would call an all-rounder, Dr. Cole earned doctorate degrees in chemistry, physics, and mathematics in France and the UK. Professor Cole is being recognized for pioneering the development of the technique of photocrystallography, and specifically for using it to develop a series of photo-excited materials. Supported by her unusually broad training, Professor Cole has applied a range of approaches to materials discovery and modification, including photocrystallography, application of molecular design rules, and the integration of numerical and ab initio modeling, as well as pioneering the application of data mining and automatic text mining to build databases. I am truly delighted to congratulate Professor Cole on this recognition, and I'll invite her to deliver the Warren Award lecture. And I'll turn the floor over to you, Professor Cole.
Okay. Well, thank you, David, for the very kind introduction. And I'd like to thank the award committee for their very kind consideration. It's truly an honor to receive the Warren Award. I can't possibly talk about everything that I've just been cited for, but I talked today about the more crystallographic side of what I do, on developing a new range of single crystal optical actuators. And most of this work is actually done experimentally at Argonne National Lab in the US, but I'm from the University of Cambridge and have a joint appointment with the UK's national source, the Rutherford Apton Laboratory. So I'll begin the talk by giving you an introduction to the scientific motivation in terms of the why we want to study these types of materials, and then tell you about the class of materials themselves, the chemistry. And then I'll talk about why, how we're really designing these materials to meet the application need, and what sort of experimental methods we use to do that. And then I'll take you through the materials discovery of a range of optical actuators and show you how we're trying to essentially control them, the parameters of them for applications. So let's begin with what are single crystal optical actuators. I guess that's a fancy name for a light-induced molecular switch or a light-induced molecular transducer, specifically solid-state ones and crystalline ones. This is quite an important slide, so I'll spend a bit of time explaining the actual chemistry of the systems because these will feature right through the talk, so it's important to grapple with these. So here is the generic chemical formula of everything I'm going to talk about. It's a ruthenium-based coordination complex. So the ruthenium's right in the center of a sort of roughly octahedral complex, and these are in the equatorial plane amine groups. These ligands will always stay the same for the purposes of this talk. And the light active part of this molecule is this sulfur dioxide group. And there's a trans-ligand, as in trans to the sulfur dioxide group X, and there's a counterion Y, and we vary X and Y chemically. Now, when you apply light to this type of complex, what generally happens is this photoactive ligand, the sulfur dioxide, can convert to one or more of these two photoisomers, right? So you can get go from the sulfur-bound SO2 to either a side-bound form, or in fact, it can flip right over. So instead of sulfur-bound now, it goes to oxygen-bound. Okay, so there are two different photoexcited states possible. And the switching process is really then this photo-isomerization. So if you imagine a dark state would be a 0, and one of the photoisomers would be a 1, so in the sort of sense of a binary switch. And all of this is happening inside the crystal lattice. That's quite important to understand. And moreover, we, as you'll see later, don't generally achieve 100% photo conversion, although we'd like to have that from applications, as you'll see. Because there's a lot of change in the configuration in these photo-isomerization processes, you get a lot of charge transfer changes. And so the crystal during this process generally is photochromic because there's a charge transfer perturbation going on. So that's the switch from a 0 to a 1. And then I also said sometimes these optical actuators behave as transducers. So I'll now describe what I mean by that. So here is the dark state of one of these complexes, to give you an example. So this is this SO2 again, and the ruthenium's in the middle with the amines, and then X in this case, the trans-ligand, is a 3-chloropyridine. And there are 2 counterions, that was Y. These are tosylate groups in this particular case. And when you apply light to this particular complex within the series, you get the photo-isomerization. So that's your sulfur bound to oxygen bound, yellow to red. And when it goes to oxygen bound, it's sticking out this, this terminal oxygen, and it's sticking out to the point where it's very close to one of the tosylate groups, the counterions, too close, in fact, for comfort. And so what happens is all within the crystal lattice, there's too much crystal strain built up by that close interaction. And so it actually, once you obtain the photo-isomerization, it's too close, the arene ring of the counterion actually rotates to alleviate the crystallographic strain. So you can see the distance now is much larger. So there's a lot of movement therefore going on. And, you know, in fact, the light that induces the mechanical change here, if you like, results in a much bigger mechanical change in the neighboring ion. So that's why it's called transduction, right? So photo-isomerization, that's the switch part, and then it's causing a knock-on effect in that neighboring ion, the transduction. And this particular example gives you 42% photo conversion. So the applications of these, some may be more evident than others, but a lot of people, for example, talking about, you know, nano cars and all sorts of molecular machines like that. And you can imagine these might equally apply to solid state devices. And so that ranges from everything from molecular motors right through to futuristic circuitry designs for quantum computers. So this is one as a design for one just here, and you see it says an optical mechanical crystal. That's a transducer here. But we've got a long way to go if we want to make these actually put into devices for applications. And so I spent a lot of time essentially trying to tailor these properties or understand these properties so that I can tailor them. And we'll walk you through these properties that I want to be able to control. The ideal is to essentially, you know, be able to have a photoisomer that works at room temperature. We'd like to have the photo conversion 100%, so it's a very clean switch or transduction. We need to understand the light that we're applying, you know, what sort of light do we need? And we also really want to be able to understand the relationship between structure and essentially the properties. So that might essentially be the optical absorption characteristics or the charge transfer changes, for example. So I'll now talk about a number of case studies where we will see that we're trying to make advances in these 4 areas. And at the end of the talk, I'll show you how far down these arrows we've managed to get. But before I show you some results, I just wanted to touch briefly upon the experimental methods that we use to obtain all of our results. The synthesis I won't say so much about, but obviously we have to synthesize our complexes. The most tricky part is what we call the photocrystallography, the in-situ light-induced X-ray diffraction. And because everything I show you will be in the single crystal form, that means we also need to do single crystal optical absorption and in fact Raman spectroscopy and also single crystal optical microscopy as well. That's less straightforward than you might maybe realize. So on the photocrystallography side, here's an example of the type of setup. I mean, this will be mostly familiar with you, of course. You know, here's a standard sort of schematic of a diffractometer, and the X-rays come in, your crystal, your calling apparatus, the detector. And when there's no light applied, of course, you just get a regular crystal structure. So, and when there's light, You may do various things to the crystal in the process, and you will then hopefully get a photo-induced crystal structure. So dark state, light state. So that's a very simplistic view, but for further reference in detail, you can see various reviews here. Quite importantly, we need to understand what you do with the data that you obtain in terms of light-induced crystal structure information. So the dark states, that's straightforward, of course, that would be the conventional crystal structure refinement. And I haven't shown the counterion in this particular case, but that would be a chloride. So this is a dark state of one of these complexes. The light-induced state, remember that there's a residual dark state, and in fact the dark state until not so long ago actually was still the predominant state. That means that we actually model our light-induced states as essentially a disorder model, right? So just in the same way that you would in crystallography model a disorder model, the light species, or possibly two photo-induced species, is modeled as the minor component or the major component and so on, right? So the occupancy factor that you're maybe familiar with when you model disorder is essentially what we call then the photoconversion fraction. So here you have actually two photo converted species. You have the oxygen bound SO2 in the dotted line, and you have the hollow lines, the side bound form. And then the black is the dark state overshadowing it, right? Because everything, of course, is averaged over all the unit cells. So it's quite important to then understand how you actually refine these data. Before you even do a photocrystallography experiments, we do complementary studies using these different techniques. Optical absorption spectroscopy is really important to us because a photocrystallography experiment will almost invariably not work unless you know what wavelength you need to probe to get the right level of absorption in your crystal so that you will actually see photo-isomerization. And here's just a picture of an experimental setup that gets made every time I go to Argonne. It has to be put together again, just to show you how it works. There's an optical table in the back there with, that's where I put various different LEDs and band passes and all sorts of things and collimation. And that goes to the crystal, which sits right in the middle of this cryostat here. And there's a microscope, as you can see, so that then shows the crystal. There's your single crystal optical microscopy. And then the spectroscopy is actually all on the same apparatus. It's actually off the left of this photo, but it's all done in one fell swoop, which is quite important to do that together. Anyway, just so you get a sense of it. But it's custom built because you have to do these in the single crystal as opposed to maybe the solution-based spectroscopy that you probably have at home in the lab. So once we've got some optical absorption characteristics, results of which you'll see later, we can know what light and what intensity and so on to apply. And we support that with vibrational spectroscopy. And again, that's light induced. And because that gives us hints of which type of photoisomer we might be expecting. And then there's the microscopy side as well. And so that will then, once you do a photocrystallography experiment, lend to the properties that we're seeking. So that's just by way of giving you a very brief introduction to the experimental methods that are applied. And you can see that a lot of these are custom made or very specialized and only exist in various parts of the world. But I now want to jump to the results and show you how we're trying to tailor these parameters so that we can try towards meet application. So I'll begin with the easier of the optical actuators to understand, which is the optical switching. So here's an example of an optical switch. This is the chemical, and so this is, as you see, the common fragment that we had right at the beginning with the photoactive SO2, and the trans-ligand here is a water, and the counterion is two tosylate groups. Okay. Nothing happens with the tosylate groups in this particular example because it's a switch, not a transducer, so I've not shown that. But here is the oxygen of the water. I'm not showing you the hydrogens. This is its start state, and you apply green light to it, and at 100 Kelvin, and you get the photo-induced state. And you see this side-bound state here has formed. And so that's the photocrystallography. And then what we do, as I mentioned, we obtain the single crystal optical absorption spectra. Now, what's interesting about this, as you can see, it's once it's photo-excited, so this is the dark state in blue, right? So that's a fairly characteristic curve. And then as we apply, maybe 1 minute of light, now a total of 5 minutes of light, then 10 more minutes. So we get 15 minutes of light and so on. And you can see eventually this saturates probably after about 60 minutes, maybe. There's not much, if you increase more time of light, you're not going to get much more optical absorption. So this gives you indications of, how much light and for how long you need to apply so that you can do a photocrystallography experiment. But you can see this, it's almost like a black body absorber by the time, because that's pretty much the full light spectrum, invisible light. So it's almost a black crystal once you get to the saturation point. And you can see this through the optical microscopy we perform. This is the dark state crystal, that's at room temperature, and we cool it down to 100 Kelvin. And then we apply, so many minutes of light to it. And after 3 hours of light, you can see it's pretty black. And the only way to change that, because that's photo-isomerized, is to warm it up, is one option. So when you go to 160 and then you, from 100, and then you wait for so many minutes, you can see that gradually it decays back, because these photoisomers exist in a metastable state. In other words, they exist as essentially forever as long as you keep the crystal held at 100 Kelvin, but above a certain temperature, they no longer metastable, so they will decay as a function of time. And see, after 3 hours, it's not quite back at the dark state, but it's getting close. And if you have that higher temperature, it would decay quicker. So one way to then go back to the dark state, it's therefore reversible, but is through thermal decay. Another way to actually get the light-induced structure back to the dark state is through applying red light, right? So we photo-excite with green, and with red, we can get it back as well. So this was the picture of green light application of for 30 minutes, and then having got it very dark, I then applied a red laser, and because it's a laser, I had a very small spot size, and I was just applying it to the top right corner here where the arrow is, and so you see this bit has already gone yellow again, back to the dark state, but this part is still very black, right? And it's many more minutes of red light application that I apply, and after an hour of red light, it's all back to essentially the dark state. Right, but what's quite nice about this actually is because you're using a really small spot size for the red light, you can see that two-tone ability I've got. So if I really wanted to put in as like a stencil, I could even write some words here, like I could write ACA in yellow and black or something like that. So this leads to sort of the idea of optical writing in terms of some application. So there's two ways to get back. Red light application, might be a bit quicker and perhaps more controlled, but warming is what we generally use, in fact, just because it's more straightforward optically. And as you saw, we've raised the temperature above the metastable temperature, and then the photoisomers decay. And we can also use that to help us understand the thermal stability of these complexes. And again, looking at the optical absorption spectroscopic data, we can play tricks like raise the, so photo-excite the material at 100 Kelvin, right? So it goes to the photo-isomerized states, and then we raise it above the metastable temperature, so that then you're going to get thermal decay, and do this and go up sort of 7 different temperatures in this particular case. So 145 K is one of them. So we're above the metastable temperature, and just at this one temperature, for example, we'll then, there it is, that we've just raised it up to that temperature. So it's just going above the metastable temperature, and then we just wait for so many minutes, right? So it's going to decay as the function of time, and we can monitor that. We can monitor that decay. And moreover, we can actually calculate the area under the curve of that decay relative minus that of the dark state. And essentially that will give us information about the rate. In other words, we can actually calculate the rate constant for this decay at the temperature 145 K. And then if we do the same type of experiment, I collect spectra like these at a slightly different temperature, like 148, 150, and 142, say, those sorts of temperatures very close by, but nonetheless above the metastable temperature, then we can actually generate enough rate constant so that we can, through the Arrhenius equation, actually obtain an activation energy of the sidebound to dark state form. So that's helpful because that tells us about our thermal stability of the photoisomer, the sidebound photoisomer in this case. So that's telling us some information about the thermal stability of the system. And another trick you can play with temperature, just so for completeness, is you can actually do the opposite. You can actually cool down the temperature right down to something like 13 Kelvin in this case. This was our base temperature for the cryostat involved. And we applied light to this material, the same material that I just showed you. And instead of this sidebound primary form, we actually obtained the oxygen bound form in two different disordered configurations, in fact. Right, so you get different states with different temperatures, and of course, it's all about the different energy levels changing. Because you know, as you see, the activation energy of from sidebound to dark state is only 30 kilojoules per mole. So that's you know, these are very subtle kind of transitions, if you like. So there's a combination then of the different uses, the complementary uses of photocrystallography and optical spectroscopy. And that tells us some things about thermal stability, say. But another thing to point out is that if you just change the chemistry, sometimes just by a minute amount, at least what feels like a minute amount, you can still obtain very big changes. So that poses quite a big problem, by the way, for molecular design. But nonetheless, I'll give you a real example. So the system I just showed you was the one in black, right? So I'm not even going to change the cation, but I'm just going to change the one substituent of the anion, the counterion, and so it was tosylate. I'm going to now remove the methyl group and put in a chlorine instead. All right, so it becomes chlorine benzene sulfonic acid, sulfonate rather. So that's it, right? That's all I'm going to change, just the counterion, right? So instead of methyl, it's chlorine in each of these counterions. Everything else was the same, and what you get is something very different. Instead of a molecular switch, we now have a molecular transducer. Right, so what's happened is instead of this, and that's because the sulfur dioxide, instead of the sidebound form that was present when we had the methyls here, we actually now get the oxygen bound sulfur dioxide forming as a preference. And because the oxygen bound photoisomer has a sort of an oxygen that kind of really juts out geometrically towards one of the neighboring counterions, that distance here is uncomfortably close. And so you get the rotation of the arene ring in the neighboring counterion to alleviate the crystal lattice strain. So just a very small change, even in the counterion, and you can change the whole crystallography, and by association, the optical properties are then also very different. So here is the optical absorption profile of the crystal I just showed you that becomes that's a transducer, because I've just changed this methyl for a chlorine. And you can see its profile here, right, is totally different from the one I just showed you where there was a tosylate group here, right? So the black body absorber, do you remember that, for the sidebound photoisomer? And because it's mostly an oxygen-bound photoisomer that forms now, it's got a very, very different profile. Even the dark state is actually quite different in this case. Yet, if you want to play another trick, you can actually not quite generate the same optical properties as the tosylate, but by a bit of trickery, this was, so what I've just showed you was the green light to green light comparison. But if I then take this crystal and with a very different profile to the one I showed you with the methyl, I can, and then I apply different light, and I do the experiment at much 100 nanometers lower, so I'm basically probing right down here, then I can not quite get a black body, right? Like I had with the tosylate. There's the tosylate, right? So it does, the drop off here is quite slight. The drop off here is much slighter, bigger, quicker, and so it comes in at a much lower wavelength. That's because I'm kicking it down at 405 instead of 505, right? So like for like, 505 nanometer pumping, these are very different. But if I want to somehow get the properties of this one to try and match that, I could play around with my lights and not quite simulate it, but kind of get close. So there's various games you can play, and then it sort of, this hopefully gives you an idea that, there's some sort of optical tunability of these systems, again, because the energy levels are very close, and these are very small activation energies involved, and so on. So that's then the first molecular transducer that I've shown you, but actually it's not the first one that was ever discovered. The first one that was ever discovered was back in 2013. We reported this material in the series, and this is where instead of water in the counterion here, we have now in fact, the halogen was chlorine, so we had 3-chloropyridine as the trans-ligand, and this was the tosylate group, the counterion being the same then, right? So the difference here is the trans-ligand. And we got 42% photoconversion. It was the first one that we ever found in this series, so it got on the cover page of Advanced Materials and so on. But this, in fact, was a repeat of the intro slide I just showed you, because it's this example here, right? So you get you get sulfur to oxygen bound forming, and it's too close for comfort, so it rotates the arene ring and loses the crystal lattice strain. Now, so this is then another transducer, and one of the control parameters I showed you with those arrows, and that what we want to try and tailor and to design better materials for applications, we really want to get this photoconversion rate up, right? From 42%, we'd ideally like 100%. So we thought about this, and we thought, well, actually this chlorine here is actually, you see it's quite elongated in its ADP, and actually there's that actually points kind of towards, I haven't got the full crystal packing here, but this is the isometric unit, but it points towards essentially the center of the arene ring in one of the configurations. So we thought, well, what if we try using a heavy halogen, right, a heavier one? So instead of chlorine, we're now going to add bromine. So we're going to have 3-bromopyridine as the trans-ligand. And what happens is now we actually do get 100% photoconversion. We've put something much heavier here. There it is in the dark state. It's wobbling a little bit. This is not representative because I've just put it. It's quite ugly if I show you the full ADP, but it's basically kind of pointing towards the if you like, nearly the center of the arene ring here that's got the disorder with the rotated arene. So, and if you look at the crystal packing, there it is above, the bromo is kind of above that. And actually, if you look at the details of the bond lengths involved, the rotor, that's the one with the rotated arene ring, the bond lengths are, quite polarized, right? So, so this is sort of almost anti-quinoidal, if you will. Compare that to the tosylate counterion that doesn't do anything, if you like, and that's much more isotropic. So you've actually got quite a polarized arene ring, and the bromo is really disordered. And actually, I'm not showing you all the results here, but and it's also photolobal, but and it's very polarized as a consequence. And so there's various evidence to show that then this is actually forming an anion pi interaction. These were, of course, interactions that haven't been known for quite so long, but have now been recognized. So this was kind of interesting, and it sort of purports to contribute to the complicated mechanism by which all of this happens. But we're thinking that these sorts of interactions are actually enabling a stability that helps towards this 100% photoconversion. I'll show you another example in a moment, which will hopefully give you more information about that. So there's the first glimpse of achieving 100% photoconversion. How does the thermal stability look like in this compound? Well, the thermal stability we can actually gauge by various means. This is the Raman spectroscopic data from we applied light at 90 Kelvin in this particular case. There it is, the 90 Kelvin structure, vibrational spectra. And this is a signature here of the oxygen-bound photoisomer. And if you then warm up and keep collecting data, so we're now at 120 in the green, and then it shifts quite characteristically here and goes at 130. It's now the side-bound form, right? So because what we do know is that the oxygen-bound photoisomer is thermally less stable than the side-bound form. And we also know that the oxygen-bound form essentially decays directly into the side-bound form, not straight into the dark state. It only goes into the dark state from the side-bound form. Right? So it goes oxygen-bound form, heated up to the side-bound form. And then if you keep heating, it would actually then go to the dark state, which is why you lose the signature at the higher temperatures. So that's the thermal stability. It's about 120 K or so. And this is the optical absorption spectra we see. This is the dark state of the 3-bromo example, and we're applying minutes, so many more minutes and minutes and minutes of light. And this is the oxygen-bound photoisomer forming actually, and then you actually get a hypsochromic shift, and there's a sort of peak here which we think is associated with the pyridile in some way. Anyway, so that's what it looks like. It's not quite a black body absorber, but it's obviously absorbing a significant amount out there. So it does dip here though, yet so this is at 100 Kelvin, and then we heated it up to just above the metastable temperature, right? So you see 130 has already gone to the from oxygen bound to side bound in the Raman. So if we go to 133.5, then we're already thermally decaying into this from the oxygen bound to the side bound form. And so what happens is this is the orange here is the profile just like we've got here. So that's when we just raise the temperature, sort of 0 minutes after raising the temperature before it's had chance to decay. And then we watch the decay. And actually what happens is you get the growth of this band, right? So because the oxygen band is decaying into the side bound, if you remember, we had the first example of that on the molecular switch. That's that kind of characteristic black body absorber, if you will. And so that's the transition there. And then if you heat it up even further, so now after some time that becomes the black body absorber, the side bound, there it is. And then what we do is heat it up even more, now way above the side bound metastable temperature, and then it will go right down to its dark state. So all of this just collapses and essentially becomes back to the dark state that you started with. So that's some indication of the thermal stability. And we wanted to then say, well, we've got 100% photoconversion in the 3-bromo. So can we justify ourselves and say, well, if we put something even heavier on, like 3-iodo, do we get the same thing? And yes, we do. We get 100% photoconversion in the iodo as well. Now, the iodo actually looks, crystallographically quite a mess. It's very badly disordered. And one of the reasons for that is actually because the 3-iodopyridine has a higher pKa than 3-bromopyridine, and the higher the pKa of a ligand in this environment, that means the poorer pi acceptor it is, right? So it's a strong sigma donor, but it's a poor pi acceptor. And what that means is that when these crystallize, if it's a strong sigma, then it can obviously, it's got nice free rotation. But if it's a poor pi acceptor, it's not necessarily gonna hold it still. So there's substitutional disorder because it's 3 substituted, right? It could be 3 that way or 3 the other way. And so actually you get some sort of, and it's very badly disordered, in both sites, in fact. But this is, I've tried to model it as anything else, and this is the only thing that we'll accept. Anyway, so that's what you see, but you still see the transduction with the neighboring counterion, but it's even messy here as well, because the 3-bromo and all the other ones I've shown you today, you know, if you like, imagine this rotation axis here, it really does, you know, rotate about those 4 atoms of the arene ring. Whereas this one, the 3-iodo, as you can see, it's actually slipped as well as rotated, right? So this is the methyl, and you can see that's you know, it's not along this axis anymore. So it's kind of rotated and translated or slipped in some way. So much more complicated, and its optical absorption spectrum is different as well. So this is the 3-bromo that I showed you just a few slides ago, and this is the 3-iodo with the same wavelength, the green light, and it's really quite different, right? You've got this kind of part here instead of this. I mean, you can see some commonality, right? So if you look at the wavelengths, you know, there's the peak and there's that peak, but it's much more pronounced here and so on. But anyway, so. This was quite helpful to know because we are also looking at the tunability of light in the 3-iodo, and this is the green light that we apply in all of the ones I've just shown you today. But if you change the light, 405 we've had once already, that actually looks quite different. If you look, see, that's the highest peak here. And now that one's the highest peak, and of course that's partly because I'm probing I'm pumping the light at that wavelength, right? So you don't get all that change, whereas you do get all the change if I probe it at 505. So anyway, so again, there's sorts of hints of optical tunability there, but the optical properties are somewhat different, and that's not so surprising because you've got different types of transduction, and it's heavily disordered and so on. Nonetheless, we can obtain an activation energy of the oxygen bound to side bound through this 3-iodo complex. And so there, if you remember, this was the idea that you use the optical absorption spectra information. You've got the photo-excited state, and then you raise the temperature above the metastable temperature to, in this case, 5 different temperatures above the metastable temperature, and then you allow it to decay. You get 5 different rate constants from the optical absorption spectra, and then you can, through the Arrhenius equation, obtain an activation energy. And I said, I think earlier, that the oxygen-bound SO2 ligand is less thermally stable than the side-bound form. You remember the side-bound to dark state was an activation energy of 30 kilojoules per mole. Well, then the oxygen-bound to side-bound, which you know is less stable, is 11, according to these results, right? So that makes sense, right? So you've got 11 to go from oxygen-bound to side-bound, and then 30 to go from side-bound to dark state. So again, we're sort of characterizing the thermal stability in various ways. So in terms of the design protocols that I've shown you through these case studies, we've now got all the way to 100% photoconversion. We've got actually now 2 examples of that. We've got some handle on the thermal stability. We've got activation energies. We haven't necessarily improved to a much higher temperature our stability, but we nonetheless understand it better. And we're starting to get some sort of ability to optically tune our systems. But the bottom one on the design criteria of those arrows I showed you earlier was that we really want to be able to understand the structure in relation to the optical absorption profiles, and actually get to sort of relate that structure property more directly. So that led us to do a somewhat different type of experiment, or more extended type of experiment. And I'm now going to show you the results on this compound. So again, the same idea, but instead of it looks like the halogen series, right? But instead of putting a 3 halogen here, I'm going to have a methyl group. That's much lighter, of course, and it's also got a different pKa and so on. And it also leads to a structure that has both side-bound and oxygen-bound. And that's quite helpful to us, because it means that if we want to try to understand the link between structure and property, we've got all of the different parameters together. Right, so here's the dark state, nothing special there. Here's the fully light-induced state, so after 2 hours of light. And this is how it looks in the packing diagram. You can see the arene rings rotated. And then what we did, so that's the fully light induced state, but we did this experiment quite differently. And so what happened is we did it in time slices. So just like I showed you with the optical absorption spectra, I had 0 seconds of light, then I had 5 seconds of light, 10 seconds of light, and so on. So now we're going to do the same with photocrystallography. So each of these frames here is a fully acquired data set and refined data set of a photocrystallography experiment, right? In fact, there are actually 12. I only put 8 on there, but there are 12 different experiments here, just in a series of applying t seconds of light, right? So obviously 0 seconds means you get a dark state. If I apply just 5 seconds of light in this particular case, you can just about refine the sulfur of the oxygen-bound photoisomer, right? You can't get the oxygens at 5 seconds of light because the population's too low. The methyl group there is starting to wobble a bit more. If I apply 10 seconds in total of light, now I can actually refine the whole oxygen-bound photoisomer, and I can also refine the transduction, so the rotated arene ring, right? And then if I apply more light, so my photo conversion is going up, so that means I can model more things more accurately, and so on. And the ADPs are starting to wobble even more, particularly on the methyl group, and so on. And then it goes up to 2 minutes of light, and we see a fairly fully developed photoconverted species now. And this is these were the photoconversion rates as a function of light duration. So there are actually 12 of them in this 2 minute time sequence. So the dark state goes down, the red is the oxygen-bound photoisomer increasing, and yellow is just the sidebound. It doesn't really go up to much, and the sort of mauve color is the rotor, the arene ring rotating, how much forms. But so that was one time sequence. Then we did a much longer time sequence, actually over about 2 hours. And in this case, the time steps was synchronized to that which I have obtained for optical spectroscopy, optical absorption spectroscopy. And that's really important because then you can put both the structure changes and the single crystal optical absorption spectra changes together, and you can literally map structure to the changes in the optical spectra. So hopefully you can see this movie. So this is now after 4 minutes, 5 minutes of light. So this is the optical absorption spectra, right? You see it's peaking up here, and then it goes down here, right? And you see things are changing in the structure all at the same time. And I've got exactly the same time points and the structural changes with each of these photocrystallography experiments as I have the optical absorption spectra. So I can also, I'm just going to play that again so we can talk it through some more. So you've also got the photoconversion rate changing because I'm modeling that with the structure. So for example, if I look at these various peaks, you see that peak it's about to top out about now, right? So where am I here? I'm just starting to converge on my oxygen-bound photoisomer. So that might be indicating that that feature, resembles something like that, right? So I can start to relate what I see in the optical absorption spectra. So dark state, that's the rise of band, that's the oxygen band forming, right? Then it's at 5 minutes, it was starting to form this kind of hypsochromic shift, right? And that's where, the oxygen band is on its maximum point and so on. So you get the idea, I can start to make literally relationships between what I see in the spectra against what I see in the structural changes. And if I put all that together, to cut a long story short, I can actually then relate, these structural features to the optical absorption spectra. And without going into all the details, sorry, it's so busy. But moreover, it's not just a case of, some sort of qualitative look at the structure. Of course, because each of these structural time points contain fully resolved and refined crystal structures, photocrystallography structures. Of course, that means I've got bond lengths of each of these time steps too. So what that means is I can actually see what's going on in terms of contractions and expansions of the bonds as the optical absorption increases, right? So if you look at the equatorial plane where you have the amines with the rutheniums, that's N1 to N4, you can see that you get the contraction of bonds. So that means that the amine rings are just contracting, going inwards into the ruthenium. right? The N5, that's the ruthenium to the N of the nitrogen of the pyridine, right? And that just collapses really quickly, and then, you know, slightly decreases after that. The yellow, that's the ruthenium to sulfur. So that was the dark state, right? The dark state's losing its occupancy, but nonetheless that increases. That makes sense because you're losing its bond strength as there's less of it, and the oxygen bound oxygen bound SO2 is forming in its place gradually. So this all kind of makes sense. But you can also see a few sort of bumps, right? And you say, well, are these real? And yes, they are, right? So at time of about 12 minutes, there's a bump, and there's time at about 26 minutes, there's a bump, and you don't just see it in the ruthenium core, right? Here's the tosylate part. And you see this, particularly C13 to C14, right? There's a massive decrease in bond length here, and then suddenly, bam, it stops at about the same time here. And then there's another little bump at 26, exactly where you see the time here. So you see these are propagating, these changes in bond lengths are propagating right through the different parts of the structure. So I then sought to actually try to understand, because this helps understand the mechanism of what's going on, what's going on at 12 minutes and what's going on at 26 minutes. What's going on at 12 minutes is kind of interesting, and it's best explained by this Hirschfeld surface. So if you look at this bit right in the blue, the black circle, so just so you know, if you can see it, so the Hirschfeld surface here, this blue blob here, that's the disordered tosylate, right? So the rotated arene ring, right? And then you've got this cation above it. So here's your methyl pyridine. Right, and remember that's sort of gradually contracting towards the ruthenium up to about 11 minutes, and then there's this kind of bump, the red there is showing a sort of small interaction between the methyl and the rotated part of the arene ring and the tosylate. So this is at 11 minutes. So watch what happens if I go 1 minute more to 12. And if you see that, I'm going to go back. So that's 12 minutes, right? So the red has gone right to the center of that arene ring. So I'm going to go backwards. It also shows, I'm just go backwards and forwards a few times, 11 minutes, 12 minutes, so on, right? You can see the methyl group has rotated, and that's changed all the interactions between that and the arene ring. And if you look at the bond distances, you'll see that actually that's alleviating the lattice strain, and that has a sort of knock-on effect on the other bond lengths. Hence, you see those increases, because of course, if this bond length changes, then the amine ligands will essentially compensate. So that's what happens at this point here, and there's a sort of knock-on effect in various points. And at 26 minutes, there's a much more subtle change, and that's actually seems to be due to the hydrogen bonding between one of the hydrogens in the rotated arene ring and the oxygen-bound SO2 that's formed from the photo-isomer, because they can only get so close, and the hydrogen bonds contract only to a certain point. At some point, they just don't contract anymore. So and three different ones around this SO2. So it seems that there's some sort of convergence limit, but that seems to happen at 26 minutes. So we're starting to see these correlations. We need to understand these sorts of things a lot more, but actually, hopefully you can see the importance of that, which is now that we have these time sequences that match optical absorption spectroscopy with photocrystallographic structures, we can start to actually assign, the changes in these optical absorption spectra, the changes in the bands, right from, the structure, rather than simply guessing them. So that will hopefully lead to much better structure property information, so we can actually start to do more kind of quantitative, larger studies. But in the meantime, let's see how we've got on. Just then to conclude how we've got on. These were our original molecular design goals. There's thermal stability, photoconversion fraction, so on. This is the time point in 2013 where we were. That's because that was the first example of an optical, the transducer, just to give you a sort of point of reference, right? And the blue, by the way, was the sidebound form. So we haven't really done much on the sidebound form, the switching, but we're working very hard on the transducers. And what I've showed you today is now results in green. So we've gone from gray to green over the sort of from 2013. So the last 8 years, we've kind of hopefully got better. We haven't managed to really improve the thermal stability. You know, the 130K is still kind of the best we can do on the, at least on the oxygen-bound photoisomer, but nonetheless, we understand all the activation energies now and so on. The photoconversion fraction, we've now got 100% photoconversion in 2 of the compounds I showed you. And the optical wavelength control, I wouldn't say it's perfect because we're still looking at tunability, but I'd say we've kind of got 90% understanding there. And just from the last example I showed you, you know, I'd say we're maybe 50% if we're lucky towards some sort of, you know, mechanistic understanding of the transduction process and trying to relate this structure to the optical absorption bands in terms of the optical properties. So that's almost everything I wanted to show you. I'm just going to leave you with a sort of, you know, if you like, a teaser of some things that may come towards in the future. This is a picture of something that happens sometimes when we don't have, it's a chloride counterion, so there's no transduction option, but there's a lot of crystal strain that's happened. And this crystal, I don't know if you can see it, but it's sort of, it's opening up, it's literally macroscopically peeling. Right, can you see it's kind of like you're peeling a potato. So this bluer sort of hue here, that's the inner part of the crystal. And just by applying light, and then when I warm it up again, actually, give you a few seconds. There it goes. It's peeled up, right, and now it's actually going to peel right back, and it almost closes up there. It sort of got cut with the light, and see it just kind of heads back. So, you know, if you don't get, you know, if you've got too much lattice strain, then it can really, you know, do even more crazy macroscopic things. So that's everything. I just wanted to thank 2 of my students who synthesized the materials, and Sven also helped one of the photocrystallography experiments. I'd like to thank Warren I've read his book and he taught me a lot about X-ray diffraction, and for funding, and thank you. Sorry, obviously Argonne National Lab, sorry, I missed them. Thank you.
Thanks so much. What an elegant talk. Thank you. We do have about 5 minutes or so we could take a few questions, and there's a couple already in the chat, which I can read if you like. Yep. So Yaohua asks, how deep does the light penetrate into the crystal? Is the light-induced structural change a near-surface effect or a bulk effect?
That's a great question. So it very much depends. The one thing I can tell you for sure, although we haven't really published any evidence for it yet, although I know internally, it's definitely a non-linear process. So if you apply Beer-Lambert's law, right, then pretty much none of the experiments I've shown you would have worked. And that's because it's not linear absorption. It's very much non-linear absorption. So there's a gradient for sure, but it doesn't just stop, right? We spent ages trying to understand this a long time ago. So you know, if you imagine the thickness of a crystal, for example, if the light only got through 50%, say, right, and not that it kind of works like this, then what you should then see diffuse scattering, because you should see that, you know, if you got 50% conversion, say, you'd expect, you know, the bottom half to be dark state and the top half to be photoexcited state, 100% kind of thing, but there's no evidence for that in terms of diffuse scattering that we would expect to see. Moreover, we do all of the optical absorption spectra that I've shown you actually are done in transmission, right? So if we didn't get the light through, we wouldn't literally be able to get the data. So we know the light is going through, but there's definitely a gradient to it. And we're sort of looking at that in various ways just now. But it is a bulk effect, but with a gradient, yeah.
Neat. And I was just wondering if there are any constraints on the changes by the fact that they're in the crystal, or would you see the same thing if it was not in a crystal? Oh, right.
If it's not in the crystal, actually what happens with these materials is they pretty much degrade within, you know, minutes. They go black, horribly black, and decompose. So they literally effectively don't exist unless they're in a single crystal.
Interesting. Okay. So Peter was asking what the size of the crystals were that you were showing.
Yeah, so the ones I've been showing, they are typically, well, if we do the X-ray experiments, we try to make them smaller. So the X-ray experiments at synchrotrons were about sort of 100 microns by maybe 50 by not a lot, maybe, 10, somewhere between 10 and 50 microns, maybe. We try to keep everything as thin as possible so that we can get as much light through as we can. The optical microscopy experiments were done, we can have them wider because there's no, we're not bound by an X-ray beam. So they can, they tend to grow larger than I just described, but we're actually looking for the smaller ones. But they generally grow quite thin in any case, yeah.
Thank you. And I see Hannah is there. Maybe you'd like to unmute yourself, Hannah, and ask your question. No, she doesn't want to. She was saying, how are the crystals grown?
Okay. They're grown straight from the reaction. Actually, they precipitate the product. They precipitate as the product, which is good and bad, because basically, if they don't precipitate, the experiment's over, and there's no recrystallization option. But they actually precipitate. It's a game of playing with your kind of acid and base mix in the reactant. Yeah.
So as a follow-up, how much are you in control? Can you I don't know, play with the temperature and make them bigger, or put the seeds or something like this?
Yeah, we try as far as we can to slow the reaction down once it kind of is getting to the point where it's precipitating. Yeah, and we can call it and things like that. That's the best we've ever managed, so we can try those sorts of things. We've not done anything more sophisticated than kind of put it in the freezer and things like, and generally try to, insulate it so that it cools down as slow as possible. Yeah.
Great. So we'll take one more because there will be a session starting soon. Margaret was asking, could there be an amphipolar compound without the need for a counterion?
Oh,
there she is.
I don't know is the answer. I've never seen one. So that doesn't mean there isn't one. What I do notice is not just in this family of materials, but in many other families of materials where people have done photocrystallography, it's invariably that there is a counterion and there are two charged ions happening, right? That doesn't mean that you couldn't do this so much on a neutral species, but if it's amphipolar, maybe if there's a stack, so they get like positive and negative in some sort of way. I don't know. I've never seen one, but that doesn't mean one doesn't exist. But I think the charge difference is actually pretty pivotal to this working. Yeah.
Terrific. Thank you so much, Jackie. What a wonderful, elegant lecture. Really appreciate it. And congratulations again on the award.